During this module, I'm going to help you revise how substances are transported into and out of cells. In this section, we shall look at the structure of plasma membranes, simple diffusion, facilitated diffusion, osmosis, active transport, and bulk transport. Remember the phospholipids with their hydrophobic tails and hydrophilic heads? The phosphate group at the head end of the molecule gives it polarity and makes the head water-soluble. So, at the watery interface between the cytoplasm and the extracellular fluid, phospholipid molecules line up in a double layer, with the heads facing outward and the tails tucked away inside, away from water. This forms the lipid bilayer that is the basis of the plasma cell membrane. Embedded in this lipid bilayer are protein molecules, some of which can move. The membrane has a fluid mosaic structure. The patterns of proteins in the lipid bilayer forms a mosaic, and the lipid and protein molecules can move, so providing fluidity. This diagram shows the structure of a plasma membrane. Some of the proteins span the entire membrane, and these are called intrinsic proteins. Some are enzymes, and some form channels to transport water-soluble molecules across the membrane. Some form ion carriers or channels. Some proteins are only partly embedded in the membrane, and so may be on either surface of it. These are called extrinsic proteins. Some of these act as receptors for molecules such as hormones. Some of them also have a carbohydrate chain attached to them to form a glycoprotein. And these can act as signals to the immune system so that it recognizes the cell as self, or as receptors for hormones and neurotransmitters. Some of the lipids also have a chain of carbohydrate added to form a glycolipid, and these also act as cell markers. They also make the membrane more stable and help cells in tissues to stick together. Animal cell membranes need to be stronger than plant cell membranes, as animal cells have no cell walls. They also contain cholesterol in the lipid bilayer, on the inside parts, as cholesterol is very hydrophobic. The cholesterol reduces movement of phospholipid tails and makes the membrane more rigid. It also makes it more waterproof and prevents leakage of ions from the cell. Cholesterol is particularly important for neurons. Have you ever been woken up to the smell of a delicious breakfast being cooked in the kitchen? The molecules from the food diffuse from where there are lots of them to where there are fewer of them. And so some of them enter your nose, dock with receptor molecules on membranes of the cells in your nose, and you know that breakfast is ready. The molecules move using their own kinetic energy, and they move randomly until they are evenly spread out, or have reached equilibrium. We say that they move from a region of high concentration to a region of low concentration, or that they move down the concentration gradient. In biology, we are usually concerned with the way that molecules diffuse across the plasma membranes. These membranes are described as partially permeable. The rate of diffusion depends on the concentration gradient. The steeper it is, the faster the diffusion. The area over which diffusion takes place. The larger the area, the faster the diffusion. The distance over which it occurs. The shorter the distance, the greater the diffusion. The nature of the cell membrane, its composition and the number of channels. The size and nature of the diffusing molecule. Small molecules diffuse faster than larger ones, and fat-soluble molecules diffuse faster than water-soluble ones. Polar molecules diffuse faster than non-polar molecules. And the temperature. Higher temperatures give the molecule more kinetic energy, so they diffuse faster. Fick's law summarizes the relationship between surface area, concentration gradient, length of diffusion pathway, and rate of diffusion. Diffusion is proportional to the surface area times the difference in concentration divided by the length of the diffusion path. Like simple diffusion, facilitated diffusion is a passive process, and relies only on the kinetic energy of the molecules involved. It differs from simple diffusion in that the molecules pass across a membrane via protein channels, or by using protein carriers. Each protein is specific to a certain type of molecule. They only open or change shape when the molecule collides with it, and then they allow the molecule through the membrane. The energy involved is still the kinetic energy of the diffusing molecule, and the molecules diffuse down the concentration gradient. Osmosis is a special type of diffusion. It is the diffusion of water molecules across a partially permeable membrane. They move from a region where there are many water molecules to where there are fewer. We need to describe this in terms of water potential. Water potential is a measure of the overall potential energy of water in a system compared with the overall potential energy of pure water at the same temperature and at atmospheric pressure. Pure water has a water potential of 0. When solute molecules are added to water, they lower the solute potential, although the solute concentration is increased, and they lower the water potential. In cells, therefore, the water potential is below 0, so water potential values in biology are negative. If water can move through a partially permeable membrane between two places, it will always move from the region of higher or less negative water potential to the region of lower or more negative water potential. Water can therefore pass from one sucrose solution to another. The more dilute of the two solutions is the hypotonic solution. The more concentrated solution is the hypertonic solution. If two solutions have the same concentration, they are isotonic. Another way of saying this is, the solution with the higher water potential is the hypotonic solution. The solution with the lower water potential is the hypertonic solution. And two solutions with the same water potential are isotonic. Consider red blood cells placed in distilled water. Water enters the cells by osmosis, down the water potential gradient. And, because the cells have no wall, they will swell and eventually burst. This bursting is called hemolysis. Consider red blood cells placed in a strong salt solution. Water leaves the red blood cells by osmosis, and they shrivel. They are then described as crenated. They lack water and cannot carry out their functions. Red blood cells placed in a 0.9% salt solution remain their normal size. The 0.9% salt solution has the same water potential as blood plasma, and there is no net osmosis between the cells and the salt solution. Let's look at the effect of osmosis on plant cells. Some onion cells are immersed in a strong sucrose solution, low water potential and hypertonic. Some are immersed in distilled water, high water potential and hypotonic. Some are immersed in a 2% solution of sucrose, which is isotonic with the onion cells. Now, if we look at the cells, we can see that those in the strong sugar solution have lost water. The plasma membrane has shrunk away from the cell wall, and the vacuole has shrunk or disappeared. The cells are plasmolyzed. The cells in distilled water are swollen, but they do not burst, as the cell wall prevents that. The cell wall exerts a pressure and eventually prevents the entry of any more water. The cells are turgid. The onion cells in the isotonic solution are neither plasmolyzed nor turgid. Sometimes molecules pass across cell membranes against the concentration gradient. This requires energy in the form of ATP. It's a bit like swimming against the tide. It also uses proteins embedded in the cell membrane. These proteins may be enzymes that hydrolyze ATP to release the energy needed, and they also may change shape and carry molecules across the membrane. Some carry one type of molecule one way, and at the same time carry another type the opposite way. All cells have sodium-potassium pumps in their membranes that do this, pumping out more sodium ions than the potassium ions they pump in. This produces a resting potential, and cell membranes have a more negative charge on the inside. This resting potential can sometimes be changed, as happens in neurons when they are stimulated. Here, we can see how glucose molecules can cross the cell membrane against the concentration gradient. Active transport helps digested food molecules, such as glucose, to be absorbed from the gut. Some substances are too large to pass through the cell membrane, so they are moved by bulk transport. Substances being moved out of a cell in this way do so by exocytosis. Secretory cells may produce enzymes, which are then packaged into vesicles by the Golgi apparatus. The vesicles then move up to and fuse with the plasma cell membrane, and their contents are then released to the outside. The movement of the vesicles to the membrane requires energy from ATP, so this is an active process. The cell membrane invaginates, folds inwards to form a trough or depression, and the substance fits into the depression and is pinched off into a small vacuole or vesicle as the membrane fuses together. The vesicle is called a phagocytic vesicle if the substance inside it is solid, or a pinocytic vesicle if the substance inside it is liquid. A lysosome fuses with the vesicle and releases lytic enzymes and other chemicals into the resulting phageolysosome. The enzymes digest the contents of the vacuole, which then diffuse through the membrane of the vesicle into the cell cytoplasm. The Protoctist amoeba ingests and digests bacteria in this way, as do some of our white blood cells. The process of endocytosis is also called phagocytosis, cell eating, or pinocytosis, cell drinking. It is an active process requiring energy from ATP.